I am Deb Meyer. I'm the Gene-ius supervisor. A few things before we start. If you were here last year at one of the workshops, it's going to be very similar to the workshop last year. I made a few changes in the problems this year to reflect some of the updates we made to the event, but it will be very similar. Again, it's really just for teams and coaches, and if a coach is a parent is a coach, they can stay, but we don't want any extra parents kind of hanging out if they're not needed. A few things too, I am going to go over sort of each section, then give the kids time to work on it. We'll walk around and see how everything's going, and then we'll get back together and go over the answers as a group. Just remember, this isn't just a workshop for you guys. It's sort of a very helpful workshop for me. It allows me to see what kind of questions the kids can handle, if there's something confusing in the way I did it, so that when I write the test, I have an idea of what's appropriate for the teams as well. Let's see. The problem set that you were given, plus the PowerPoint, and apparently a video of the presentation are now going to be posted following this, so you could go back and look at that as well. I will be posting some updates and clarifications after this workshop and the workshop I did a couple of weeks ago, as well as a few updates to the study guide. I found a few minor things in there. There was a picture that was covering some things. Anyway, a few cleanup items, so I'll be posting that updated study guide this week as well. Let's see. All right. So again, today we're just going to kind of focus on those more problem-based aspects of this event, the things the kids will be asked to do during the event that are not the multiple choice or the true false, those kind of standard questions. So DNA structure, codon reading, karyotype analysis, Punnett squares, pedigree analysis, and some of the blood group genetics problems. Okay. Just another clarification. They will not get all of these at the event. The event is only 30 minutes. They will not have time to do all these problems. So 4th grade and 5th grade will both be given a subset of these, as well as a traditional type of test format as well. So again, we're going to kind of review the concepts just very briefly. They'll do the problems, and then we'll go over them together. So we'll start out with DNA structure. I want the teams to be familiar with both what is represented in the model that they've been given, as well as if they were given a picture like this in the event. They need to know the basics about that it's a double-stranded helix, that each strand is composed of stacking nucleotide, what a nucleotide is made up of, a phosphate, deoxyribose, and one of the 4 bases. They need to know the base pairing rule, A to T and G to C. They do not need to memorize the colors of the bases in the model. During the event, they were given, if they use the model, they'll be given a little color code card so that they don't have to memorize what they are. They need to know the anti-parallel nature of the double strand. This year we changed that. Both 4th graders and 5th graders need to understand that the 2 strands run in opposite direction, and instead from head to tail, which we did last year, we just went with the actual proper nomenclature of 5' and 3' end, with the phosphate end, a free phosphate end being called the 5' end, and the free sugar end being called the 3' end. They don't need to understand why it's called 5'. They need to understand the designation. And again, if you're looking at the model, it would look like this. A properly assembled model will look like this. Now, the instructions that came with the model are actually incorrect. So when you got your model, it would have had a little clarification thing that I printed out, an extra step showing that in order to correctly assemble your model, one end should have the phosphate, the purple part, and then the other end would have the free sugar. And then if you could see this, the end of this one, if you followed it down, would have a free phosphate. They'd be swapped on the other end. Okay? So in case you lost that little supplemental instruction in your model, just want to make sure you understand that. For now, I want you to go ahead and work on the first question, which is just page 1. And if you don't have your model, you won't need your full model, and you won't be actually assembling it, because they won't need all the pieces. That would be great. If you didn't, don't worry about it. You can do it at home as well. Again, I'm trying to think of creative ways to use this model than the traditional way, because there's no way during the event they could do the whole thing. So again, this is one of those things where asking you to do that, building the model, helps me understand what you guys are capable in a certain amount of time and whether it's something that's feasible in the event. Obviously, this model is a great model, but it takes some time to put things together. So the first question is quite easy. It's just what are the 3 parts of a nucleotide? Okay, so you guys, it's phosphate, deoxyribose, and base. Any order is fine. The second one was to build, and that's not a great picture here, but I also have one here. So again, I was asking for just a single strand, so you wouldn't have been forming it on your model, because that would have to be a double strand. And this would be the order. Again, you have the free phosphate on one side is your 5' end and your 3' end. This way, actually, too. Doesn't matter as long as you formed it the right way. Okay? I'll leave it up here, and you guys can do that as well on your own. Okay, so the last question, again, this is just to clarify your understanding of this anti-parallel nature, understanding that you guys, what I'm asking for when I use certain terms. So what would be the sequence of the complementary strand of the above sequence? So in the worksheet, it was the one above, which is that first one you see there. What is the sequence in the 5' to 3' direction? So my thing isn't working very well. Again, that would be this way. So if you flipped it, you'd be giving it to me in that direction. However, I would have accepted this as long as you designate which side is the 5' end and which side is the 3' end, okay? If you just give me a sequence without this, then I don't know which orientation you're talking about, okay? Does that make sense? Okay. All right, so the next activity that they're going to need to know is how to utilize that codon chart to read gene sequences and translate it into a protein sequence. So again, when you're talking about a gene is a section of DNA, some of which contains the code that will be used to generate a certain sequence of amino acids and then a protein in the end. And then the proteins are what is important to go on and do all the functions a cell needs to do. And so that DNA sequence is read as a triplicate, 3 bases at a time corresponding to a specific amino acid. For the 5th graders this year, they need to understand that going from that gene sequence to the protein or amino acid sequence goes by way of an intermediate RNA, another nucleotide, nucleic acid, and that the process that DNA is converted to an RNA is called transcription, and then that process where the RNA is actually read and converted into an amino acid sequence or protein is translation. So 4th graders don't need to know that. 5th graders need to understand that this year. Okay. So this is the codon chart that you have. And again, they need to know that they need to read them as triplicates and then how to use the codon chart to distinguish which amino acid is being designated with that triplicate code. The other thing I want to mention, too, is that they should start reading a sequence at the first ATG. For our purposes, we will always say that our sequences are going to start with ATG, because that's mostly what is used, and then read from there. very often when I give you a sequence, because I cannot give you a whole entire gene sequence, it will be a partial sequence. Okay, but I will give you a sequence that will start. You look for the first ATG and read from there. Okay. They need to know a few things about mutations. And so a mutation is really any change in that gene sequence from what is commonly found there. They don't always have negative effects. Sometimes they don't do anything. Sometimes a one nucleotide change will still code for the same amino acid because of something called redundancy in the code. Don't worry about that. But sometimes it does make a big difference. And sometimes you can get a change that will cause a different amino acid to be placed in the protein, which is the case you see in sickle cell anemia, where it's just a one mutation change that changes glutamic acid amino acid to a valine. And that one amino acid change really makes the protein very different and function different and cause sickle cell disease. Oh, the other thing I want to mention too is also instead of just amino acid substitution, sometimes it can put code for now what's called a stop sequence, and so now you have a shorter protein. And the types of mutations that they should be familiar with for this event are missense mutations, which is a single nucleotide change resulting in amino acid substitution, so now a different amino acid would be placed in that position, or a nonsense mutation. This is the instance where you get now a stop codon being inserted there. They should also understand a frame shift mutation. This would not be just a change, but an actual insertion or deletion of a nucleotide, shifting now what's called your reading frame. So you're going to get everything downstream of that is usually altered. Okay? Okay, so you guys should do the next 2 pages, I think, here. Yes, page 2 and 3, and then we'll go over it together. The question is about a partial sequence to the dystrophin gene. And this is just a little bit more additional information about dystrophin, but you don't really need to know that to answer the question. So go ahead, and 5th graders, you do page 3 as well. And even if you haven't finished, you can always go back and finish it up later, but I want to keep going because we have more things to do. So the first page, page 2, the first question is to look at that sequence and using the codon chart, give me the amino acid sequence. Again, I want to show you that if I write ... that means there's more to come and we just don't have time to keep going ourselves. So I want everyone to understand that. There were a couple of questions and I just want to clarify. I do want the full word for the amino acid and not the abbreviation because I don't want kids to be losing points by getting those abbreviations wrong, especially for certain amino acids like glutamine and glutamic acid that are very similar. So please write the full one, but spelling will not count as long as I know what you're writing. Okay? So if you guys quickly look at yours, you'll be able to tell if you got it right. And again, you needed to start your reading at the first ATG in the sequence I gave you. Okay? Oops. Sorry. Okay, so the next question was to look at this new sequence I gave you and identify the mutation, which was right here, a T instead of a G, and then write the amino acid sequence down for this new one. So you'll see it should look like this, and then yes, it should be the same except for when you get to now what is now a stop codon, okay? So what type of mutation is this? This would be a nonsense mutation. And when I ask this question, what effect will the mutation have on this dystrophin protein? Anything of this nature indicating that you understand you're going to have a short protein, a smaller, non-functional protein, something, because it's so small, will be appropriate. Okay. Again, the PowerPoint will be posted online as well as the presentation. Okay? So the next page was really just for the 5th graders, and it's just filling in the blanks that I left there. So here, to put the template strand in, you're going to be doing the complementary sequence. This is to indicate transcription, and now you'll have your RNA, which will be the same sequence as your coding strand, except now instead of T's, you have uracil or U. Okay? Translation is this process that takes the RNA into an amino acid sequence, and this should be your sequence that you should get from that strand. Okay? So that was for 5th graders to do. All right, we good? All right, so the next type of problem you might be would be interpreting karyotypes. This one is usually pretty easy for everybody, fortunately. So chromosomes are packages of DNA in the cell. And you need to know that humans have 23 pairs, 22 autosomes, and a set of sex chromosomes, either X or Y, or 2 X's and an X and Y. This is a very colorful picture of a particular karyotype. But what a karyotype is a collection of the individual chromosomes from one cell that a scientist will isolate, and they kind of look like this when they first do it. And then they arrange them in pairs, starting with your largest chromosomes to your smallest autosome, and then they put the sex chromosomes at the end, your X and Y. And what they can do when they look at a whole set of your chromosomes is identify whether you're male or female. Also, large changes in those chromosomes, missing chromosomes, extra chromosomes. The kids don't need to understand translocations and things like that. We're just looking at the large scale things. So they should be pretty comfortable looking at a normal karyotype and then looking at an abnormal karyotype and being able to distinguish what the difference is. They will only get this type of karyotype, a single chromatid form, on the test. If you're looking for karyotype samples to show them on the web, you often see the kind with the X. I won't be showing that kind. I'm going to try to be consistent and always show this type. Okay, so the next problem, page 4 and 5. 5 is your questions, and they refer to the karyotypes you see. The 5 is a picture of the karyotypes, which are these up here. And the questions are on page 4. So this won't take very long. This is pretty easy. Just take a few minutes and look at those really quickly, and we'll go over them. Okay, is everybody ready to go over these? Because we have some other ones coming up. They're going to take a little bit longer. All right, so karyotypes are relatively easy to look at. You just, oops, I'm sorry. Once you get familiar with looking at them. So again, which individuals are female? That would be A, here, 2 X chromosomes, and B. D. D. Those are the 2 that show 2 X chromosomes. Which are male? That is B and C, because they have the X and Y. Right. How many chromosomes are present in a normal human? 46. And are any of these karyotypes abnormal? C has 47. It is Down syndrome. You can see right here. And D is trisomy 13. You can see it right here. I would also accept Patau syndrome in the event. So just to clarify that, there are 2 genetic defects, Down syndrome and Turner syndrome, that are listed in the study guide that I expect them to know the names of. So I would expect them to give me Down syndrome in the event because I'm looking for them to actually know Down syndrome specifically and Turner's. They're the only 2 that they should know by name. All the others, they can give me trisomy whatever or monosomy whatever. If they do know the correct genetic syndrome and they give me that, they will also get credit. So I'm not going to deduct points for them knowing more, but if they have an expectation to know something and they don't give it to me, then they will get a point deduction. Okay? Just to clarify that. Okay. Let's see. Now, this is something that they need to spend a lot of time on understanding the terminology here. This is usually very daunting for them to understand all the words and have a really good command of the use of those words during the event. It'll be very important. And again, they need to know when I use the word gene, we're talking about a particular segment of DNA that is responsible for a specific trait. In this case, we're talking about coat color, fur color in dogs, these dogs, okay? Allele is referring to different versions of that gene. In this case, we have a black allele and a yellow allele. They need to understand dominant. You only need one copy of that allele, and you will see that trait being expressed. Where the recessive, you would need 2 copies of that particular allele. They need to understand what genotype means. Genotype is actually your combination of your alleles that you have, and your combinations could be homozygous dominant, in this case 2 big B's, homozygous recessive, 2 little b's, which would mean 2 of the yellow allele, or heterozygous, 1 of each. And that it's the genotype that will then, and I'm sorry, it's a little dark here. I hope you can see it, but it's your genotype that will then distinguish what trait or what your phenotype will actually look like. And again, whenever you have 1 copy of the dominant allele, you will have that phenotype or trait, but the recessive phenotype when you have 2 copies. So they really need to understand all that language and have good command of it. That information is useful then when they need to set up Punnett squares, which they will likely be given in the event. In this case, we're talking about the alleles for, I think it's rat eye color here, with black being the dominant allele and red eyes being the recessive alleles. So if they were told they had a parent that was homozygous dominant for black eyes, that would mean that they had 2 alleles for black eyes, and they would be putting them on one side of the square. And if they were homozygous recessive, they would have the 2 alleles for red eyes, and they would be put on the other side of the square. Then they fill in each square, which will show you the possible combinations for offspring. And each one of those squares represents 25%, potentially 25% of your offspring. Okay? They need to be able to give me the percentages of expected offspring or fractions. A fraction I would accept as well. Okay? So you need to know how to set these up. It does not matter which parent they put on each side. That doesn't matter at all. Another clarification, which was asked at the earlier workshop, I'm going to give you the letter I want you to use when you set up these things. Last year, I got a large variety of letters being represented, some of which are very difficult to tell the difference between. A big C and a little c written by a 5th grader very quickly can be very problematic when I'm grading. So I am going to tell you what the letter I want you to use will be, and I'll try to pick things that are easy to distinguish uppercase and lowercase for. Okay? Now let me see if I have any more here. All right, so you guys should do the questions for page 6 here, and it's about free earlobes, which here I'm telling you to designate as the capital E, and attached earlobes is the recessive, so that's small e. Okay? So go ahead and work on those problems, and I'll come around. Okay. Hello? All right, so let's go over these quickly. First question says, what are the genotypes of this couple? If you have a heterozygous free earlobe male, that if it's heterozygous, you know he has to have 1 of each. And if it's a female with attached earlobes, again, just the key that she has attached earlobes, you know she must be homozygous recessive. Right? So that allows you to determine those. And then using the genotypes, you'll fill in your square. It should look like this. Okay? So using your combinations in the square, answer question 3, which is what are the possible genotypes of the offspring and the expected percentages or fractions. And so you should get 50% that would be homozygous and 50% that would be homozygous recessive. Okay. What are the possible phenotypes of the offspring? Again, make sure you understand what I'm asking for, whether I'm asking for genotypes or phenotypes. In this question, it doesn't matter. We have the same answer, 50% free earlobes, 50% attached earlobes. But sometimes it is different, and so you have to make sure you're paying attention to that. Okay? Everyone good? All right. So the next thing you're going to need to do is pedigree analysis. These tend to take more time when you're working on them. So a pedigree is a way for us to look at the pattern of inheritance of a particular trait. Say you just want to look at something like eye color or something like that, or a pattern of inheritance of a particular disease. And it's often called a family tree. Okay. I want the teams to be really comfortable identifying the components of a pedigree chart, what is represented by a square, a circle, a filled-in square or circle. What lines attaching mean, which is a mating or a marriage. That lines coming off that pair represent their offspring. All right. Also, this half-shaded square would represent a carrier. And most of the time, you do not know if somebody is a carrier or not. So they need to understand that when you see an open circle or a square in a pedigree, you really can't just tell whether that person is a carrier or not. I might ask you, and I think there's some sample questions in here where I ask you, for you to tell me based on the information that you're going to fill in the chart, whether you can tell if somebody is a carrier or not. Okay. Sometimes you can look at a pedigree and you can figure out the pattern of inheritance based on the way it is set up, and other times you cannot. For example, in this case, this is showing, even this very small little family here, is a dominant trait. And you know that because it can't be recessive. Because if it was recessive, this father and this mother would both be carrying 2 copies of that recessive. That means all of their children would have to have it. In this case, not all their children have to have it. So it obviously can't be a recessive trait, so it has to be a dominant. In this case, it has to be a recessive. How do you know that? You cannot have a dominant child where you had a parent who didn't have it. Okay? So that one has to be recessive. In this case, this particular small pedigree doesn't provide you enough information. It could be either dominant or recessive. So that's why typically you're going to get larger pedigrees with more family information to help you identify what you're looking at. But these little key components, often within a larger pedigree, will help you. To work on and answer the questions. The one on page 8, it says 5th grade only, but I think the 4th graders can answer most of those questions anyway. So go ahead and work on those, and then we'll go over them together. 10 and 11. I will accept, if asked this question on the event, anything that indicates that they know what their relationship is, either siblings, brother, sister, 2 brothers, things like that. Those are all fine, acceptable answers. Same thing with 3. What is the relationship between individuals number 8 and number 9 here? Married couple, husband and wife, mating pair, whatever it is that indicates that they understand the relationship. How many generations are represented? Basically, each line represents a generation, so 3. Which individuals have hitchhiker thumb? 5, 7, and 12, because they're the filled-in ones. What is the most likely pattern of? Okay, so number 14 is, it could be either of these, because these parents are obviously both heterozygous. Okay, they don't have, number 14 does not have hitchhiker thumb. So they obviously have, you don't know if they have 1 or 2 alleles for, based on this. If this was the kind of thing you could always, if you know what the parents are, you could always do a little Punnett square in that case and figure out what are your possible combinations. But does this, are we good? This year we introduced X-linked recessive for 5th graders. And that's why I tried to clarify this year using autosomal recessive and autosomal dominant to distinguish from X-linked recessive for So I hadn't really thought about that. Probably for a 4th grade test, I would accept just recessive. For 5th grade, I wouldn't, because of the fact that 5th graders also have another new one, that pattern of inheritance we introduced this year. Okay? Okay, the next one has to do with color blindness. I put this in here because you guys can all take a turn and see whether you think you're colorblind or not. If you can see the numbers, you're not colorblind. Okay? All right, now that we're all done looking at that. Okay, so this next pedigree chart here, again, I'm going to ask some of the same questions. How many generations? In this case, we got 4 generations. And then I asked, in this pedigree, do you see more males or females? And you see, obviously, the people affected with color blindness are predominantly male. So then I asked, what is the most likely pattern of inheritance? Most likely pattern of inheritance. Again, if you see a recessive trait that is much more proportional in males, X-linked recessive is going to be your most reasonable answer. Okay? All right, so the last question, oh wait, did I ask another? Oh yeah, identify all the carriers. I'm sorry. So based on what you know that it's an X-linked recessive, if you went back, you should be able to identify carriers as such, and the only ones you would not be able to know for sure were 19 and 20. Okay? I'm not going to go over every individual one right now because we don't have time for that, but that's what it should look like. Okay? Again, females would only be carriers as well with an X-linked recessive. Okay, this will be posted, so I'm going to move on here because we have another thing to do. So the last question is about blood group genetics in humans. And this year, 4th graders and 5th graders may be asked questions about it. This is the only example of multiple alleles and codominance that they will be expected to know for the event. You're going to be working with multiple alleles A, B, and O. Codominance, because A and B, when are present, will both be expressed, and that gives you the AB blood type. And they're both dominant over O. For this event, they will need to know, they won't be given a chart like this, so they'll need to know what genotype has what phenotype. And when you talk about phenotype, that we're talking about the actual blood type of that individual. All right, they'll need to know that. They'll also be asked to designate the alleles as such, just with A or O, not little I with an A or B, just because it gets really sloppy, again, when I'm trying to correct these things that the kids are writing. So that's how I want it designated in the event. All right? Both the genotypes and the phenotypes designated with A's and O's, A, B's, and O's. Okay. So the last one, you should go ahead and start working on it. I think I have it. Okay. This is an example of something I want them to be able to do. If given a written family tree, they need to be able to use that information to correctly construct a pedigree. All right? So it takes a little practice, but they should be able to do it. The order of siblings does not matter, okay? Again, I don't care the sequence those siblings come in, as long as they're represented correctly in the family. Okay, so you guys do the last one and then we'll go over it. It takes a little time to go over, so. Alrighty, so based on the information that was provided in my little description of this family, you should set it up like this. This is what you should get just by using the information that's written in there. Should look like this, okay? Now, based on this information, we should be able to fill out all those other people's blood types or phenotypes plus everybody's genotypes. So first, we're going to do the genotypes of these individuals because they're really easy to identify. Because if you have blood type O, it's your phenotype, your genotype has to be OO. And if you have blood type AB, then your genotype has to be AB. Okay? So those are the easy ones we're going to do right away. We're going to work our way through this. based on that information, we are going to do these next individuals. So if you have an individual who's blood type A, but they have a child that's O, then they had to donate an allele O to that child, so they have to be AO. Okay? And that would be the same thing with this individual here. They had an O, and they are A. Here, they have to be AO as well. Well, not necessarily. We'll get to that one later. But A and B here, same thing, because they have a parent who is O, so they obviously have to have an A because their blood type is A, and then the O from that parent. Okay? Let's move on to the last individuals. Again, if you have an individual here that is an offspring that's O, and this person is blood type B, they have to be BO. Same here. Okay? If you have two children that are one's A and one's B, and your spouse is O, then you had to donate that A to one child and the B to the other child. So that's what that is. Okay? So this takes some time to fill out the whole chart. But it's a good practice in taking a written family history and filling in the whole chart. Okay? I would never ask this many questions or this big of one in the event because it would take too long. But, okay? So that is it for today. I'll stick around to answer any other questions. We're going to be posting this, both the worksheet, the PowerPoint, and the video of today's for you guys. Okay? Thank you.